I'm sorry for this delay. So if you can let me know if you can hear me in the chat, please. Okay, so I'm just gonna wait a few more minutes. Okay, hi guys. Can everyone hear me in the chat or no? Okay, perfect. Is it lagging or no? Is it good? Is it better now? Okay, perfect. So I'm just gonna continue while everyone else joins in. Okay, so till now, what did we say? We said the enzymes are made up of proteins. Okay, their function is they assist in chemical reactions. How do they assist in chemical reactions? They do that by speeding up the reaction. And an also important thing about enzymes is that they do not get used up, so they do not get consumed by the reaction. That's why we call them biological catalysts. And as I gave you the example, we have our substrates or reactants on the left side while we have our products on the right side. So this reaction, for example, maybe could take 100 hours for it to occur. However, once I insert an enzyme or a catalyst, this reaction can maybe be completed in 20 or even 10 hours, okay? That's everything we learned about till now, and I think none of you have any questions. So if you have no one has any questions, let's go and look at the structure of the enzyme, what binds to it, where does the substrate bind, and all of that, okay? So I'm going to draw, for example, an enzyme, okay? So this is going to be my enzyme. Now, what do we call this in red? I call this the active site, okay? The active site or the catalytic site. I think in the book they mention, okay, catalytic site, okay? They don't call it the active site, I think here. They call it the catalytic site, or they even call it here active site, okay? So they're both similar, it's the same thing, okay? That's where the reaction occurs basically. Now, this site has a specific shape. I can have another enzyme, okay, that has, yes, active site is the catalytic site with the binding site, perfect. I can have another enzyme, for example, that is shaped like this. Okay, this is more of a square shape, this is more of a oval shape kind of, okay? It's like curved. So those two enzymes have different active sites. Why do they have a different active site or a different binding site or catalytic site? They're all together, okay? Because we have a substrate, okay? We have different substrates, okay? Now, each enzyme is what then? Each enzyme has a specific substrate, okay? So we don't have one enzyme that works on every single substrate that we have in our body. That won't be efficient. It's more efficient if I have different enzymes. This is one enzyme, I have another enzyme, and I could also have another enzyme. They all have different binding sites. Why do they have different binding sites? So they're able to bind to different types of substrates. So this is my substrate, okay? And this is also my substrate, okay? Now what's going to happen? This substrate is going to be what? Is going to bind here. Why will it bind here or not here? The drawing is not perfect, okay? But bear with me. Imagine it is perfect. This shape is complementary to this shape, right? And the box shape is complementary to this shape. That's why this substrate will bind only to the enzymes that have this binding site. That's why we said they are specific, enzymes are specific, and this substrate will only bind to enzymes that have this binding site, okay? So look what they say in the book over here. They're saying that an enzyme acts on what? On a substrate. Of course, after the reaction, so once the substrate binds to the active site, we have a reaction, and then we release products, okay? We release here products, okay? So the molecule, the substrate, is then converted with the help of the enzyme into different molecules called products. And enzymes are typically much larger than the substrate, of course, because if I'm going to have a larger substrate than my enzyme, then how would the reaction work? Well, the enzyme is the one helping the reaction. It has to be a larger molecule, okay? That's why usually it's larger and called the catalytic site. Now, what is this catalytic site? It's located near the binding site, and it is the site at which the enzyme, the binding site is the site at which the enzyme binds its substrate. And what is the catalytic site? The catalytic site and the binding site together form the active site of the enzyme, okay? And as I told you, the enzymes are what? Are specific to their substrate. They bind to the chemical and they catalyze. So see, the enzyme that normally acts on one substrate will not act on a different substrate, okay? Now, till now, everything's understood or no? If everything is good, please let me know in the chat before I move on. Okay, perfect. Now the theory of binding. Okay, so didn't we say that this substrate is gonna bind to the binding site, and the active site is made up of the catalytic site and the binding site, and this substrate will bind also. Now, how do they bind? Okay, so what is the process that happens when this substrate binds here and when this substrate binds here? So we have two theories. Okay, so we have two theories according to how this binding occurs. So how does this binding occur? We have the lock and key theory, okay, and we have the induced fit model. Okay, now the induced fit model is the one that's usually accepted by scientists now. Okay, that's the one they usually go with. The lock and key theory is an older model, which they're not really accepting of it. They more go with the induced fit model, but I don't think you have to be aware of that. But just extra information for you guys, for whoever's interested. So from the name, lock and key theory model, what do you expect? It's very simple. The name explains it. So it's as if you have a lock. Okay, so for example, this is a lock. And then I have a key. Okay, and it's not the best key, guys, okay? This key will directly fit into the lock. Same thing at your homes, in your rooms. If you have a door, you have a lock in the door, right? There's only one key that's going to enter that lock. I can't use the house key for my room door, right? They have different locks. That's why I need a different key. And the same thing happens in enzymes. Look here how they explain it in the book. So the lock and key theory, they're telling you the active site is like a lock. Okay, it is specific, like the door lock, like the lock of a chest, for example, like the lock of a closet maybe. Okay, so each object has a different type of lock. It is specific. Not every key I'm going to put in. I can't use the door key for a chest key because they have different locks, so they have a different key. And they're telling you they have a specific substrate that fits like a key. So what do you mean that it fits like a key? That means they are complementary to each other. Okay, for example here, this shape is complementary. Okay, this shape completes this shape. This shape completes this shape. When they complete each other, we call them, they're complementary. Okay, so meaning that the shapes like the substrate and the active site are what? Are complementary. This word is very important. Okay, are complementary. And then they tell you that when the enzyme molecule binds to the substrate, they form a temporary enzyme substrate complex. Okay, so how does this work? Like I explained here. So you have the substrate, okay, and you have the enzyme, and you have the active site of the enzyme, which consists of the catalytic site plus the binding site. Okay, now this substrate will fit in like a key. This is the lock, the enzyme is the lock, the substrate is the key. It fits in perfectly. Now they're bound to each other. Once they're bound to each other, they form a molecule called, okay, it's a temporary molecule. We call it, very simple, enzyme substrate complex. Why do I call it like that? Because it's made up of the enzyme and the substrate, and they're together, so it's a complex. Okay, then after they bind to each other for a temporary period of time, their action is completed and we have products are released. Okay, and look, as we said, the enzyme remains intact. Why does it remain intact? We said because enzymes do not get used up in a reaction. Okay, and that's it. Why? Because they are biological catalysts. Okay, that's basically the lock and key theory model. So the lock is the enzyme, the key is the substrate, and they are complementary to each other. And here is just an example of how this works. And remember, the enzyme speeds up the chemical reaction by bringing these two molecules together. And the same thing here. Again, you have the enzyme, you have the active site. It fits complementary to it. This is the lock, and this is the key. They fit to each other. What do we call this? We call this enzyme substrate complex. And then we release our products. And look, the enzyme, we can use it again in the next reaction. So they do not get used up. Okay? Did everyone understand the lock and key theory model? If everyone understood the lock and key theory model, please let me know in the chat below. Okay. Jana said no. Okay, Jana, what don't you understand? Let me know so I can re-explain which part or just in general. Okay, let me just speak about it again. Okay? And let me know if you don't understand or not. So, the lock and key theory model. Think about it like that. Don't you have a lock in the door? You have maybe a closet that has a lock. You have maybe a chest that has a lock. Okay, you might have a safe that has a lock, correct? They all have different types of locks. Locks are what? Locks represent the enzyme. So the enzyme, look at it, it has a specific active site. Okay? This active site is not the same for each enzyme. It's like the example I gave you here. This active site is not the same as this. So for example, those are two different locks. For example, this can be your door lock. This could be the lock of a safe. Now the key I use to unlock the door, do I use it also to unlock my safe? No, it's not gonna work. Why it's not gonna work? Because the shapes are not complementary. It's the same concept in enzymes and their substrate. I can only use the door key to unlock my door. And I can only use the safe key to unlock my safe. Here it's the same concept. This substrate, its shape is complementary to the active site of the enzyme, and this substrate is complementary to the active site of this enzyme. So they basically fit like a key and a lock. The key and lock, they fit each other perfectly. So this, for example, this key cannot unlock this because it has a different active site. And this substrate or key, because the idea of it, the lock represents the enzyme, that's why I want you to know, and the key represents the substrate. Okay, did you understand a bit better now? Okay, perfect. And the rest we're all good with it, right? So I can move on to the induced fit model. Now the induced fit model basically builds on the lock and key theory. Now what changed a bit? Look what changed. So here they tell you it extended the lock and key theory. So the lock and key theory was first, after it came the induced fit model. What does it say? It says that the active site of an enzyme is not initially completely complementary to the substrate shape, as proposed in the lock and key theory. So what happens in the induced fit? What does it say? I can have an enzyme. Okay, let me redraw it again. I can have an enzyme and I can have a substrate. And this substrate is not perfectly fitted for this active site. Now if I follow the lock and key theory, I say this reaction will not work. However, what does the induced fit model say? The induced fit model says that the active site, which is right here, is not completely complementary to the substrate. But what happens once the substrate binds to the active site a bit, the active site changes its shape. Exactly, the enzyme will make itself complementary to the substrate. Perfect. It will change its active site, small modifications, not major modifications. So it's not gonna go from something like this to something like, for example, a square. Okay, this is a major modification. It doesn't go for major modifications. It makes a minor change just to make the substrate complementary, to make its active site complementary to the substrate. So here, look what it says. It says, instead, the binding of the substrate to the active site of the enzyme induces structural modifications. So we have a change in structure, but minor, which changes the active site to make it complementary to the substrate. And then again, they will form an enzyme substrate complex once they bind to each other, and then they will release it into products. Okay, so it's usually you have substrate plus enzyme, they bind to each other, they form an enzyme substrate complex, and then they form the products. Okay, did everyone understand the two theories of how enzymes and substrates bind to each other? We talked about the lock and key theory, and we also talked about the induced fit model. Okay, all good till now? Okay, perfect. Now the way we name enzymes. Naming enzymes is very simple. Okay, you just look at the substrate and then remove the end letters of the substrate and add ASE. Okay, that's how most of them are called. So for example, proteins, I'm going to remove the end and add ASE, so it's going to be proteases. The ones that break down lipids, I remove the end, I add ASE, lipases. And breakdown of carbohydrates, we call them carbohydrases. because we need a water molecule. That's why we call them hydrases. While other enzymes are more specific. So the ones that break down starch, we call them amylase. But look, the ones that break down maltose is going to be maltase. Ones that break down sucrase. You also have enzyme that break down lactose. Who's gonna tell me the enzyme that breaks down lactose? What am I gonna call it? Someone in the chat, try. It's very simple. Just remove the end and add ASC. What will the enzyme that breaks down lactose be? Lactase, perfect. It's going to be called lactase. Okay? Perfect. We're all good with it? This is the induced fit. Okay? This is just a figure for you to see. So you see the active site is not perfectly complementary, but once it binds, the enzyme changes the shape slightly, not major modification, slight change just to make them fit, and then we'll have the enzyme substrate complex, and then you're going enzyme substrate complex, and then you're going to have the products. Okay, perfect. Now we have two types of enzymes. Okay, very simple from its name. Intracellular. What does intracellular mean? Inside the cell. So intra means inside, while extra outside. Okay, very simple. So all enzymes are made inside the cells. Most enzymes remain inside to catalyze their reactions in the cytoplasm and the nucleus. What do we call them if they're inside the cell? We call them intracellular enzymes. Okay. While other enzymes are released outside of the cell, what do we call them? When the enzyme changes its shape, does it go back to its original shape? Yeah, like when it releases the products, look at it. It went back to its original shape right here. Okay? So you see this shape and this shape, it goes back to its original shape. And then again, once the substrate binds, it slightly modifies its active site so the substrate can perfectly bind to it. Okay? No worries. So the other type of enzymes said are extracellular, which are released outside the cell. And they also gave you some examples. Okay, it's good to know the examples of extracellular and intracellular enzymes. Okay, now let us look at the factors that affect enzyme activity. So in the first live session, the one that cut before, what did I tell you guys to know? To know this very important sentence: structure affects function. Okay, this is very important to know. Now we're going to be looking at it. Why? So what are the factors that affect enzyme activity? So we have those factors. I need you guys to know those factors: temperature, pH, the concentration of the enzyme, and the concentration of the substrate. So we have four main factors that affect the activity of the enzyme. We have temperature, we have pH, we have the concentration of the substrate, and we have the concentration of the enzyme. Okay? Perfect. Those are the four main factors. Now let's look at each factor by itself. First, we're going to be looking at temperature. Effect of temperature on enzyme activity. So usually, what did we say? When we increase temperature, what's going to happen to the molecules? Their kinetic energy will increase, so the reaction generally increases as we increase temperature. Look, here they told us that most chemical reactions occur faster at higher temperatures. Why? Because the reacting molecules have more kinetic energy. However, okay, that's the same thing. Since they have more kinetic energy, they're moving faster, so they're going to bump into each other more frequently. And an enzyme is more likely to bump into its substrate at a higher temperature than at lower temperature. And we already said why. Because temperature increases the kinetic energy. What is kinetic energy related to? Related to the speed of the molecules. Okay? So we have, let me show it to you. For example, I'm not gonna draw full enzymes, but say black is enzymes, okay? And you have blue are substrates. Now those are moving, everything is moving very slow. So for this enzyme to move over here, it's taking time to move. However, if I increase the temperature, then I have an increase in kinetic energy. So I have an increase in velocity or the speed. So now those are rapidly moving over there and those are rapidly moving over here. So since they're all rapidly moving towards each other, they're more likely going to bump into each other, form enzyme substrate complexes, and release products. Okay, that's what happens when we increase temperature. However, we cannot increase the temperature to a very high temperature. Some enzymes cannot work at high temperatures. Some enzymes, we say they get denatured. Now, why do they get denatured? Again, they do not work anymore. Why? Because high temperature will affect their structure. And once the structure of an enzyme is disrupted, what will happen to the function of it? No more function. Okay, so that's why I told you guys structure is really important for function. If I put a really high temperature, I can disrupt the structure of my enzyme. Once I disrupt the structure of my enzyme, I'm going to affect the function of it. Okay? And here what they tell you. So what's the maximum temperature? It depends. So there's no one specific number. No, it's not necessarily to be 60. You have different enzymes, and each enzyme has a different temperature at which it gets denatured. That's why, what do we say? We say enzymes have an optimum pH. Okay, so some enzymes, look, let's look at the book what it says. So some enzymes work best at a particular temperature, and what do we call the temperature at which it works best at? We call it optimum temperature. So I'm gonna write it here. Here is it. Each enzyme has its own optimum temperature, the temperature at which the enzyme works at its best. Okay, so the best temperature at which the enzyme will work at is called the optimum temperature. Now this varies. Some enzymes function best at high temperatures. Some enzymes function best at a lower temperature, for example, maybe 30 degrees Celsius. Some enzymes function really good at 60 degrees Celsius. So it depends on each enzyme. And we said for an enzyme, if I increase the temperature too much it cannot handle, I will get a denatured enzyme. What's a denatured enzyme? It is non-functional and can no longer catalyze any reaction. And here they give you an example. Digestive enzymes work at 37 degrees, while plant enzymes work better at 28 and 30. Okay, look, you even have enzymes that work at 75 degrees Celsius, which is hot. So it depends on their environment, it depends on their function, it depends on several things. So there's not one specific temperature, it varies from one enzyme to another. But what you have to know is each enzyme has an optimum temperature. What's an optimum temperature? It's the temperature at which it works best at. Okay? And here they're telling you the activity of human enzymes increases with increases in temperature up to about 40. Now this is very important. Okay, guys, they always ask about this. I promise you this will come in your exam. Like I'm 99% sure this is going to come. I'm 99% sure it's also mentioned in the quiz because it always comes. So in general, a 10 degrees Celsius rise in temperature doubles the rate of a reaction. Okay, you have to memorize it. So if I increase, usually, if I increase the temperature by 10 degrees Celsius, I expect my reaction rate to double. Okay, you must know this. Okay, that's what you have to know for this. And here they're telling you, yeah, it's not difficult. Okay. However, above 40 degrees Celsius, what will happen? Their reaction will begin to decrease. By about 60, all the enzymes stop because the enzymes have been denatured. And look, this is the very important sentence that I told you about before. At high temperatures, the shape of the enzyme is changed. So since the shape or the structure of it is changed, it cannot perform its normal function, thus preventing the active site from forming a perfect fit with the substrate molecules. Therefore, we will have no reaction. Okay, so here also are other examples. That's just examples. This is a graph, an important graph to know. So look, at 40 degrees Celsius, look at the rate of my reaction. It's highest at that point. So what do I call this point? We call it optimum temperature. Now, once the reaction rate starts to decrease, here it's negative, it's a negative slope, so it's decreasing. The enzyme is slowly becoming denatured until we have a zero reaction rate. We say the enzyme is completely denatured. Okay? Did everyone understand how temperature affects enzyme activity? If everyone understood, we can move on to how pH affects enzyme activity. And it's also the same concept. Okay guys, temperature and pH, very similar concept to each other. Okay? Yes, usually it's irreversible. Okay? Sometimes, if it does not reach to a zero reaction rate, so it's not been 100% denatured, we can have some enzymes that are partially reversible. But I don't think for your guys' level you need to go into that amount of detail. But if you guys have learned it and know it's irreversible, let's take it as it's irreversible. Okay? Okay, let's look at the pH. I think everyone understood the temperature. So the pH is the same concept here. But before we talk about how pH affects, what is pH? pH is a way to measure acidity. Okay, so it's a way to measure acidity. It measures the concentration of what? Hydrogen ions, so H plus ions. Okay, that's what pH measures. And it's usually the range of numbers is from 0 to 14. Okay, so we have 0 to 14. We have 7 in the middle. 7 is neutral. 14 and above is basic. And then, so 7 up to here, we call it basic. And from here to here, we call it acidic. So the smaller the number, the more acidic the solution is. So now if I ask you guys a question, answer me in the chat, please. I tell you, you have an acidic solution. Do you have a high concentration of H plus or a low concentration of H plus? Yeah, if in the quiz it said it's irreversible, take it as irreversible. Okay? But who can answer my question? Okay, my question was, okay, let me repeat the question. If I say you have a solution that has a pH of 3, does this solution have a high concentration of H plus or a low concentration of H plus? Okay, we're getting some answers. The rest, if you want to join in and answer so I can say which is the correct answer. Okay, so we have 2 people that said low concentration and 2 people that said high concentration. Can we have one more person? High concentration. Okay, we have 3 versus 2. The 3 people have 1. It is a high concentration of H plus. Why? Because what did we say in the beginning? We said pH is an indicator of the concentration of hydrogen ions. Okay? Exactly. Low pH equals high H plus concentration. Perfect. Why? Because what did I say here? A low pH, so here a low number of pH, what does it signify? Signifies an acidic solution. So when is a solution acidic? A solution is acidic when it has high concentration of H plus, okay? Or we can say a low concentration of OI minus, of OH minus hydroxide. And a solution is basic. When is a solution basic? If it has low H plus and high OH minus. Okay? So here, solutions with a high pH have a lower hydrogen ion concentration and are considered to be basic or alkaline. Typically, potable water has a neutral pH of 7. So we said, as I told you here, 7 is neutral pH. Okay? And how do we measure this? We measure it by a pH meter. An example is litmus paper. Okay? Now, does pH affect the property of proteins? And what did we say enzymes are? Enzymes are proteins. So, of course, pH will affect enzymes. Again, most enzymes work best at a particular level of acidity or alkalinity. Again, we have an optimum pH. The same way we had an optimum temperature at which the enzyme works best out, we have an optimum pH at which the enzyme best works out this pH. Okay? For example, pepsin. You'll learn it later on. If you guys are interested, I have a video on the digestive system. It talks about pepsin also, which is a type of enzyme that degrades proteins. Now, this enzyme is found where? In our stomach. And our stomach, the pH in our stomach is 2. Our stomach is acidic. Okay? Okay, we have a question. Can you repeat the concentrations? Okay. For example, we said pH. What does pH do? Measures the concentration of hydrogen ions. We have the ranges are like this: 0, 14, and 7. pH of 7 is neutral. Anything above 7, we call it a basic solution. Anything below 7 is an acidic solution. Now, how do I measure? I measure using a pH meter. Okay, so measure using pH meter. An example is litmus paper. Okay? Now, if I have an acidic solution, so for example, pH of 2, that's an acidic solution. In an acidic solution, I have a high concentration of H plus ions. Okay? In a basic solution, so a high pH, for example, pH of 9, I have a low concentration of hydrogen ions. Yeah, lemon juice is less than 7 because it's acidic, right? It's going to have a pH lower than 7. And if you guys usually look at a water bottle, if you read the back of a water bottle, you're going to find the pH. So I have a water bottle right in front of me. The pH is 7.6. So it's not perfectly neutral, but I can consider it nearly neutral. It's 7.6. It's not high. It doesn't have high alkalinity. Okay? So you can check it on a water bottle you have. They also mention the pH of the water. Okay, here, and the pH of our body, just a note for you guys. The pH of our body is usually neutral too. That's why, look what they say. The optimum pH for most enzymes in our cells is at or near neutral pH 7. Because we want it to be working in our cells and our body's pH 7. We don't want it to work in an acidic environment, because then our body will have to change its environment. And we don't want to get into those issues. Okay? Okay, sure. I'll try to do the quiz as fast as possible. Okay? I want you guys to pay attention here, because if we know the material here, I'm sure the quiz will be way easier. Okay? Okay, so here it's just a graph. Look, at pH around 2, we have optimum pH for pepsin. But at pH around 7, we have optimum pH for amylase, which is another enzyme. So you see we have two different enzymes, and they have two different optimum pH. Okay, we're nearly done. So we spoke about temperature, pH, how they affect now, the concentration of enzymes, and the concentration of substrates. Now, what do you guys expect? If I increase the concentration of substrates, what will happen to my reaction? Okay, so my question was, if I increase the concentration of the substrates, what do you expect the reaction rate? What do you expect will happen to the rate of the reaction? Will it increase? Will it decrease? Will it stay the same? Or does it depend on something else? Increase, perfect. But now there's a small note that I have to add to. Again, let me give you an example. Black is going to be enzymes, blue is going to be substrates. Okay, so I increase, let me just erase some, okay? I have 4 by 4. 4 enzymes and 4 substrates. If I increase the concentration of substrates, okay, they're more likely going to bind or going to interact with the enzymes, correct? Correct, because I have more substrates, I have a better chance for them to interact with each other. However, now, if I keep on increasing the concentration of the substrates, but I keep the concentration of the enzymes the same, but now I have this interacting with this interacting with this interacting with this interacting with this. All my enzymes are full, but look how many substrates I still have. Now, does this affect my reaction rate? No, because my enzymes are fully occupied. So that's why if I look at the graph, I'm moving to the right, the concentration of the substrate is increasing. So look what's happening to the reaction rate. It's also increasing. But look over here. Over here it stops and then it becomes stationary. Why does it stop over here? It stops over here because all my enzymes have been fully occupied. So even if I increase the concentration of my substrate, my reaction rate will remain constant. Did you guys understand this? And it's the same thing when I increase the concentration of the enzymes. Of course, if I increase the concentration of the enzymes, so I have more enzymes, now the substrates can interact with these enzymes and the reaction rate will increase. Okay, let me repeat it. What part did you guys not understand? Or do you want me to repeat it all? Okay, let me know again if you did not understand or not, because all please. Okay, that's fine. Let's look at it again. Okay, now I'll explain it. Before I talk about the increase in the concentration of substrate, let's look about the... Okay, yeah, that's right, high. Okay, but let me just explain it again for everyone too. Okay, thank you for supporting them also. But let me just do it again so everyone can understand it even better. So before I look at the increase in concentration of substrate, let me look at the increase in the concentration of enzyme. So once I increase the concentration of enzymes, okay, I'm gonna draw it again. Remember, black is enzymes, blue is substrates. Okay, so we have 6 enzymes, we have 6 substrates. Now this reaction is not going to be super fast, but it's also not going to be super slow. It's going to be average, okay? However, I want to increase the rate of my reaction. So what am I going to do? I'm going to increase my enzyme concentration. Now these substrates have a higher chance to interact with an enzyme. Why? Think about it. If I just had 3 enzymes and 3 substrates, okay, they're going to interact, but it's not going to be as fast. However, if I have 10 enzymes and only 3 substrates, of course these 3 substrates are going to find an enzyme faster because they're more spread out, so I can easily find another enzyme. Did you understand this part? When I increase the concentration of my enzymes, the rate of reaction will increase. Let me guys know please in the chat if you understood what happens when I increase the concentration of my enzymes. Okay, perfect. Now I'm going to be looking at what happens when I increase the concentration of my substrate. Now pay attention. I have 3 enzymes and I have 3 substrates. Okay, again, the reaction is not going to be perfect, super fast, super quick. It's going to be normal, average speed. Now I increase my substrate concentration. And look, as I increase my, so for example, this could be 0, 5, 10, 20, 15, 20, 25. Okay, so I'm increasing my concentration of substrate. Look what's happening to the rate of reaction. It's increasing also. Okay, why is it increasing? Because I have more substrates, so the probability that they react with an enzyme is higher. I have 10 substrates and only 3 enzymes. Of course, these substrates are going to find an enzyme quicker than if there were only 3 enzymes. Correct? Did you understand this? Let me know in the chat, please, so I can move on to the next point. Okay, perfect. Now, if I keep increasing my concentration of my substrate, okay, I'm still increasing, but it was already high, but I'm just increasing still. Now, I already have a high probability of my substrate binding with my enzyme because I only have 3 enzymes. So let's say those 3 substrates, okay, interacted with the 3 enzymes available. Okay, now all my enzymes are full. I don't have enzymes that can extra enzymes for all of those remaining substrates. So what's going to happen to my reaction? This is the point where we call my enzymes have been saturated or fully occupied. So once they are fully occupied, even if I keep on increasing my substrate concentration, nothing will happen to the reaction rate. Why? The reaction rate is determined by what? By the rate of what? Of products. Okay, reaction of rate is determined by what? Products per unit time. How do I get the products? When the substrate interacts with the enzyme. But all my enzymes are fully occupied. So if I keep on adding more substrates, but I don't have an enzyme so the reaction can work, so it's going to reach a constant rate. It's not going to increase anymore. The only way I can make it increase again, who's gonna tell me in the chat, what's the only way that I can make the reaction increase again? If you understood this concept, you'll be able to answer. They don't have to be on an enzyme, but if you think about it, I already have 100 substrates and only 10 enzymes. The probability that they interact with each other is also high already. So if I add another 100 substrates, 200 substrates, and only 10 enzymes, but the probability of them binding with each other in the beginning was already high. So me adding more substrates, my enzymes are fully occupied. My reaction will be constant. Exactly. If I add more enzymes, so I'm going to add more black dots, more enzymes, Now these substrates can interact with another enzyme, and that's how my reaction rate will increase. Perfect. Did you guys understand it now? The effect of, remember, temperature, pH, concentration of enzymes, and concentration of substrates. All good. Okay, if we're all good, let's look at the final part, which is enzymes and biotechnology. Okay, so what is biotechnology? Biotechnology is defined as the exploitation of biological processes for industrial purposes. Okay, so you have to know the definition. What is biotechnology? Now, how does this happen? We use enzymes. Okay, so we use enzymes in pharmaceutical companies and also in food industries to the production of biodetergents and biofuels. You don't have to know what they are, but for many medication, many pharmaceutical companies, they use biotechnology to produce certain chemicals. They use that using enzymes. Okay, that's why enzymes are very important, and we can play around with enzymes, okay, in biotechnology. Now we have one example of biotechnology. It's called recombinant DNA technology. It's used for the production of the hormone insulin in the pharmaceutical company. Okay, so usually what do? They get a bacteria. Okay, so can you imagine, they get a bacteria, they insert an enzyme inside of it, okay, and they make this bacteria start producing insulin. Because some people who have type 1 diabetes, I think it's type 1, do not produce insulin in their bodies. So pharmaceutical companies, what did they do? This is called the biotechnology field, okay? They insert enzymes in this bacterial cell, and those enzymes are programmed to produce insulin. So we start having bacterial cells producing insulin. Okay, so this is just an example. And also here, biological washing powders, also biodetergents, they also contain two enzymes, protease and lipase. Who's gonna tell me what protease breaks down and what lipase breaks down in the chat? Anyone can help me? Okay, protease breaks down proteins, exactly, and lipase breaks down lipids. Perfect. هلا amino acids is broken down by peptidases, but like, okay, you can think about it also in a similar way. They all work out. And yes, you guys are 100% correct. And here they tell you protease helps remove stains caused by what proteins? Because protease works on proteins. And they give you example. Blood has proteins, milk has proteins, egg has proteins. Okay? And also lipase breaks down lipids. Okay? And you also have production of biofuels. Which enzyme they use for the production of biofuels? They use cellulase, which breaks down cellulose. Okay? And we can also use enzymes for what? Production of cheese. Okay? So those are just examples of where we can use enzymes and what industries they're used, and some examples. Okay, so they're telling you, no, the use of enzymes has many commercial advantages. They are specific, as we mentioned before. They only allow specific reactions to take place. They are cost effective. Why are they cost effective? Because only minimal amounts of them are required, and they produce products of consistent quality. Yes, cellulase breaks down cellulose. Perfect. You just look at the substrate and then you add ASE at the end and you will figure out what the enzyme is. Okay. that is it. Okay, if you guys have any questions, let me know. I can explain them before we end the live. Regarding the quiz, I'm gonna try to go live again in maybe like 10-15 minutes. Maybe I just need a break, but it's also because I have my university studies. So if I'm not able to go live to solve the quiz with you, send me the questions and I'll answer them. I'll try my best to answer them all if there's anything confusing. But I hope this session helped you all, made some concepts clear. If anyone has any questions about what I spoke about in this session, let me know. No worries about it. Shahid, if you have any questions, send them to me or send them on the group, and I'll try to look at them, all of them, and answer them. 3.4 is just practical work. I think you guys can just look at it. It's just equations and experiments that they did. Okay, so I don't think it's that important. It's self-explanatory probably. But if you have any questions about it, let me know if it's in the quiz or something, and I'll do my best to help you guys. Okay, so thank you guys for joining in and watching. I hope the session was helpful. For anyone who's new to my channel, don't forget to like and subscribe and comment. Please support the channel. And thank you so much again. Best of luck on your exam, and see you soon, guys. Thank you all. Bye bye.